Welcome everybody. It's my great pleasure to welcome Towlson to our in-seminar for this series. She comes all the way from Calgary. She's in the computer science department there. Also has an appointment in physics, astronomy, full member of the Hotchkiss Institute, as well as the Alberta Children's Hospital and the Complexity Neuroscience Science Group, pardon me, and part of the Computational Neuroscience platform. She'll talk to you about her work. What I thought was actually also quite impressive was this additional connection with an organization called Homeward Bound, which is a value-driven global leadership program that aims to change the narrative around leadership and promote the impact of women in science on decisions that shape the future of our planet. There's a website actually dedicated to that, so if you go to Emma's website, you can get a link to Homeward Bound and learn more about that there. It's quite an impressive organization. But we're here to talk about other things as well, so let's give her a chance to talk about networks, enters of vulnerability to brain dysfunction. Welcome, Emma.
Thank you so much for the kind introduction, and thank you for the invite. Maybe I'll stay sitting with the way the screens are. But yeah, the Homeward Bound network that you mentioned, just if anyone wanted to chat about that, I'm very glad to, especially anyone who identifies as a women scientist. There'd still be a couple more cohorts being welcome to create a network of over 1,000 women. So if that's something you wanted to chat about, please do. But yes, the main topic of my talk is network signatures of vulnerability to brain dysfunction. I'm coming from a more computational angle, from the network science angle. So if there's something that you'd like to talk about a little more, I understand we've got a lot of different backgrounds here. Please do just interrupt and ask me. I'm going to spend a lot of the time talking about the intuition and what things mean, so we can dig more into the technical stuff offline if people would like. So a little bit about me again first. I am not speciesist in my approach to network neuroscience. I look at a lot of different organisms on a lot of different scales, from C. elegans at the level of neurons and synapses, through Drosophila, also neurons and synapses, the mouse, where we start to look at collections of hundreds to thousands of cells connected by bundles, human brain, trying to understand how the architecture of our brain influences, especially things like our risk to brain disease, disorder, and mental illness. And recently, a little bit of work into the architecture of artificial neural networks as well. And so today, I'm only going to be talking about the human brain component. Just if anyone's interested to talk about any of these, I have a lot of diverse interests in this area too. So the brain as a network, we've collectively as a community been studying this for probably approaching 30 years now, and we've learned an awful lot about the way it's organized and what we think this organization means for function. We know that the human brain contains a lot of network architectures such as modules, so groups of brain regions that have denser connections between the group, within a group and between them. Rich clubs, so a densely connected set of hub regions. These are brain regions that have many more connections than others, but they seem to be densely interconnected with each other, and they seem to bridge these modules. It's thought that this organization supports integration and segregation of information in the brain, so we can have locally segregated processing that is then connected together through the rich club. We're looking at the level of the whole system. This organization is also thought to support a balance between economy and efficiency. So the brain is not optimally wired in the sense of it's not the minimal wiring. There is a cost to a lot of the wiring that is observed in the brain. But when you analyze this from a network perspective, you get a lot for where it seems to invest the cost. You seem to get a lot of bang for your buck. So a high efficiency from where the high cost edges are. And a lot of these are through the rich club. So we've learned a lot about this and what the healthy brain looks like. We've also started to learn a lot about what the brain looks like in dysfunction. So what happens when these architectures start to see alterations and disruptions, and how these relate to various different conditions. So for example, if the modularity is changed, do we see fragmentation in the modules, or reduced or increased connectivity within or between them, say is the rich club diminished or enhanced, and how this relates to different pathologies. So some examples here, depression. There's not a perfect agreement in the literature on depression, but as a network disorder, there's some findings that are consistently coming out. We find an elevated connectivity in default mode network and attentional networks, and reduced connectivity within reward networks, cognitive control, which appear to correlate with particular symptoms of depression, such as dysphoria, rumination, anhedonia, and disruptive cognitive control. There's a lot of also recent works subtyping depression into different connectivity patterns and seeing that the extent of different modules, such as the salience network. Schizophrenia, we also know changes network structure. So this is from one of my earlier papers from 2019, showing that we know in schizophrenia that clustering is reduced, but so is path length. So the brain of people with schizophrenia appears to be more efficient, but also less clustered. So you've got this a kind of trade-off of one thing that seems to benefit information processing almost, but short circuits across different modules, so it disrupts the structure. This also responds to medication, so this structure can then be altered back towards something that looks a bit more like the healthy brain with medication. Alzheimer's disease, and this is an area that I think a lot of people in the room know an awful lot more about than me, and I'd love to hear your perspectives on some of my slightly related work to this later in the talk. But we also know a lot about Alzheimer's as a network disease. We see that system segregation appears to decrease both with age and with Alzheimer's, but in a differential way within Alzheimer's. So segregation is decreased in Alzheimer's. There's some conflicting evidence about hubs. There seems to be a conserved architecture of the hubs, but also some diminishing of the weights of these connections. So we're starting to get a big picture of what these various brain diseases, disorders, and mental illnesses look like in individuals. But by the time we develop some of these conditions, it's too late. So by the time we've developed an illness such as depression or anxiety, you're not going to get the optimal outcome. You might not be able to be perfectly able to reverse it. You will get a much better impact if you intervene before you get the diagnosis. For dementia, the best available treatment we still have healthy eating and exercise. And by the time people get a diagnosis, this has been progressing for years upon years. We need things that are able to tell us that a person is vulnerable to these conditions before they have actually manifested, before we get the clinical diagnosis. So I'm going to talk about today a couple of projects from my lab that are contributing in this area to try and understand vulnerability in the brain to various conditions. So specifically two projects, one looking at the impact of ovarian hormones on the brain and their relation to affective disorders. Second one, transient ischemic attack and cognitive decline. And then a little about the outlook of the use of network control theory as a framework to explain some of this and as a potential predictive tool for interventions. So the first project about ovarian hormones and vulnerability to affective disorders is motivated by the observation that women are twice as likely as men to suffer from affective disorders such as depression, anxiety, and PTSD. Interestingly, this difference in rate emerges at adolescence and declines post-menopause, which is a pretty compelling argument for ovarian hormones being a driver for the difference in risk of developing the two disorders. So when you have a look at levels of hormones across a menstruating woman's cycle, you see something like the following graph. The black line is estrogen and the blue line progesterone. And you see how these rise and fall within the cycle. You have a point where they're both low in early follicular. Estrogen starts increasing to a maximal around ovulation, then this dips. And then both hormones are elevated during the mid-luteal phase, with progesterone also. And there's a hypothesis that in this phase, where both hormones are elevated, that there is a window of vulnerability where we're not saying that women are suffering from affective disorders at this point, just that they are more vulnerable to them. If they were to experience an adverse event in this moment, you are more vulnerable to being kicked into a place that maybe you could develop an affective disorder and more sensitive to these things. So we collaborated with a team in Boston who collected brain imaging data on a group of healthy menstruating women. 31 participants were measured at 3 different points in that cycle. So you're seeing on the, oh great, we can see my mouse here, on the graph over here, divided into early follicular phase, where both hormones are low, late follicular, where estrogen is elevated but progesterone is low, and then mid-luteal, where both are elevated and progesterone more so. We also have actual hormone measurements from most of the women, so we're not just relying on putting people in particular phases. Results in the literature have been a little mixed, and we think this is partly why of a vague definition of phase rather than literal hormone measurements. So we have both. In terms of complete data sets, where we have the hormone data and 3 phases for one woman, and we have 20 of these complete cycles. Yeah, so like I said, we have a unique hormone profile for each woman, and scans at 3 phases. The scans we have are fMRI. So we have bulk time series, which we then create functional networks out of. The functional networks are derived from Pearson correlation coefficients that Emily Fisher said transform. On these networks, we then try to recover these modules that I mentioned earlier. So we use community detection algorithm that's based on the Louvain community detection. I'm going to comment a little bit more about that in a moment. We set out to maximize modularity. What this means is we're trying to maximize the number of connections that exist within each community compared to across them. And importantly, we let these things change. So we don't impose this structure firstly on every single woman. Each woman can have a different structure within their network, a different modular breakdown, and secondly, throughout the phases. So we let these modules change their components, change their structure throughout the phases of the menstrual cycle. We incorporate both the positive and negative edge weights, and our network began with 1,000 cortical and 54 subcortical nodes. When we do this community detection, we bring together the network science measures, one of which I've already mentioned, and then pair them with what we already know from neurobiology. And this is largely following the process in the paper I referenced here. So from the network science side, what we want is, like I said, a greater density of connections within our communities compared to across them. We also want the partition that we retrieve to be stable across repeated applications of community detection. So this is something that we have to tweak parameters, and we don't want tiny tweaks of these parameters to lead to large differences in community breakdown. So we check for both of these things. We sweep across a large parameter space and check that it's stable. We check that there's a high modularity. And that is from the purely network world. We then fold in what is already known from the neurobiology world, which is we have an expectation of roughly what some of these networks should look like. We don't think our breakdown is going to be any good, for example, if it doesn't have a visual network or if it doesn't have a default mode network. So it should roughly correspond to known functional networks, and so roughly the same number and roughly the same constituents. We also want to see a separation of primary sensory motor subnetworks, so visual, auditory, and somatomotor. And then a third one that's more specific to our study, not from this paper. We don't expect things like the visual network to be affected by our menstrual cycles. That would be a bit problematic. So we are expecting a great deal of consistency within visual and auditory subnetworks across the menstrual cycle. So given all of these criteria, we then run through our process and find different community breakdowns for each woman that then vary through their menstrual cycle. We then roughly match the communities we found to known resting state networks. So we reference the Yao et al. Paper, which has a canonical breakdown of 7 functional networks: visual, somatomotor, dorsal attention, ventral attention, limbic, frontoparietal, default mode. We also have a subcortical network and an auditory network. So we have 9 networks per woman that then fluctuate across the cycle. We modified an algorithm popular in computer science to create a matching between these to automate the process, so we don't need to manually do everything here. So this is an adaptation of something called the Gale-Shapley algorithm that then allows the matching between the communities. This is then a representation of what we found. Let me break this down a little bit for you. This furthest on the left-hand side is representing the early follicular phase. So this is both hormones are low. The middle one is late follicular, so estrogen is high, progesterone is low. And then mid-luteal, where they're both elevated but progesterone higher. This is a representation of the entire cohort. So these are community structures that are represented across all women, rather than individuals. So this is then kind of an average representation, but from individual brains. Each of these blocks then represents a functional network. So this red block is the default mode network, blue block somatomotor, purple visual, then frontal parietal, dorsal attention, ventral attention, limbic, auditory, and then this is subcortical. Highlighted the networks that we're most interested in for their relation to affect and to memory. There's a few things to notice of how these change. So each of these branches, this means that a group of nodes is fragmenting off this network, the frontal parietal, and moving to default mode. And then say a branch down here is default mode, so something fragmenting to ventral attention to dorsal attention. So there's some of these branches that are much larger than other branches, and these are things that are consistently observed across women. And these things are as highlighted. So this switching of nodes from frontal parietal network to default mode, dorsal attention also, and ventral attention. So we have these attentional networks moving towards default mode in late follicular. And then the default mode fragments in late follicular phase to the mid-luteal phase with regions moving back to the attentional networks largely. So what's specifically interesting as well about some of these? Firstly, this difference is much earlier than we were expecting. The window of vulnerability hypothesis that I mentioned to you is later in the cycle, so mid-luteal when progesterone is rising. So this is really suggesting that there is a role of estrogen as well as progesterone changing brain connectivity. The specific regions that are involved when we drill down into that, instead of just looking at branches between the large resting state networks, if we pick out regions that are moving between these branches, we find some key players that our collaborators have pegged as playing an important role in especially emotion regulation and negative affective processing. For example, posterior cingulate cortex, we find that it increases its connectivity with the default mode in late follicular and mid-luteal phase. So if you look at these box plots over here, interaction strength is telling us about the fraction of connections that exist between these regions and the whole network out of those that could exist. So the higher it is, the more connectivity there is between them, the lower, the less there are. And you'll see that under the influence of low amounts of hormones, the interaction strength is fairly low at 0.42, and then we see this big increase as hormones start to rise. So that's in the phase level. When we then take this to like out of the discrete approach and move it to more continuous, so try and find a relation between this interaction strength and the literal hormones, we find that there is a relation with progesterone. As progesterone increases, so does interaction strength, not with estrogen with this one. We also find anterior cingulate cortex, which is something that often comes up, especially associated with depression. This also changes connectivity with default mode network, this time later in the cycle, more with mid-luteal phase. But we still seem to find that this is a progesterone-driven effect when we look at linear mixed effects models. And we also find relation between brain regions that have been implicated with major depressive disorder and other affective disorders, and their connectivity with default mode. So middle temporal gyrus increases its connectivity again in late follicular and starts to decrease by mid-luteal, and this seems to be a much more estrogen-driven effect. So we're able to start teasing apart these specific region-level differences with entire networks that's coming at it from a purely data-driven approach and then weaving it together with the neuroscience. We find evidence that ovarian hormones are changing the modular architecture of functional networks throughout the cycle. The alterations occur much earlier than expected, so suggesting that estrogen plays at least as much of a role as progesterone. But yeah, we find complex mixes of both. The modular alterations, and this is ongoing work, but qualitatively we're finding that they're similar to those observed in depression. So what is often described as connectivity increases and decreases in specific networks, a lot of this is a perspective thing of at what point do you say that something has reduced its connectivity enough for the nodes to just fragment and be part of a different network. So we're coming at it more from that angle of allowing the fragmentation to happen and then comparing this to literature that's got hypo and hyper connectivity in these functional networks. We see a lot of qualitatively similar things with the connectivity of our networks, which is a bit scary, but yeah, just qualitatively the same. So the second study then is on TIA and vulnerability to cognitive decline. And this is one where I think everyone in the room will be ahead of me on this, but a transient ischemic attack can be thought of as a mini stroke. So this is a transient ischemia, so a place where we have inadequate blood supply to brain regions. We often find that people who suffer a TIA go on to suffer a full stroke, and even if they don't, they are at a much greater risk of suffering from cognitive decline. 5.2% of afflicted individuals are diagnosed with dementia within 1 year of the incident, accelerating onset of dementia by an average of 2 years. So this appears to be a mediator of cognitive decline and a window into early cognitive impairment. So as a population, there is an opportunity for us as researchers to understand what makes them more vulnerable to cognitive decline later. They don't suffer from that immediately, but we know they're more vulnerable. And so this study is in collaboration with one of my colleagues at the HBI, Phil Barber. He had a study together, the design as follows, where we scan a bunch of people baseline. So this is where you've got a group of people who've suffered from a TIA, a group of people who haven't, but are controlled same age, etc. And then scan them at 1 year, 3 year, and 5 year. This is now structural data, DTI. Okay, wait a second here. The problem with COVID happened in the middle of this. I think a lot of people suffered with this, so there's quite an attrition rate, sadly, in this study, that we have many less people in the later years than at the beginning. That is what it is. It reduces statistical power somewhat, but yeah, it is what it is. We still managed to have around 30 participants that made it all the way through to the 5 year from 150, so baseline in each group. Average age, the ages are between 50 and 75. So I mentioned in Alzheimer's disease, there's some evidence that hubs are affected, but largely that the connectivity within the group of hubs, the Rich Club, seems to be mostly preserved. We started by taking a look at the hubs to see if we could find any difference, to see what the Rich Club looked like. So we measure this by degree, so just the number of connections that a node has with other nodes. We do this in a weighted sense, so we sum up the strength of these connections. To get the Rich Club, I'll show you a little bit more about that in the next few slides, but we pick out the regions that are then more densely connected with each other. But just looking at weighted degree and their distribution across the brain, we find that healthy controls and patients, so TIA sufferers who remember are subclinical for cognitive decline, we find that the distribution of weighted degree is incredibly similar, largely indistinguishable. So we do still see that you have the same kind of, at least spatial distribution in terms of degree, and we do still have the hub nodes. We then drilled down a bit into what the actual Rich Club is. What we're looking at here in these figures is look at the blue lines to start with. The blue lines is giving us a measure of the prominence of this Rich Club. So the normalized coefficient is telling us the density of connections within the club compared to those that could exist, and then corrected for chance. So the red line is our real data, the green line is comparable random networks, this is our baseline, so the expectation for a given degree distribution, and then the blue line is the normalized. Every time this is above 1, there is evidence for a Rich Club behavior. So this panel A is a baseline, so the control group versus people who just suffered a TIA, and then this is the 5-year follow-up. So everyone seems to be exhibiting a Rich Club behavior. We have this increase of the coefficient where it grows larger than 1, which is good. We do seem to see, at least visually, that at the 5-year follow-up, the TIA group doesn't reach as a greater height as before. So let's come into that for a moment. After 5 years, it's also observed from other physical measurements that atrophy in the TIA group is more than twice that of controls. And we believe that it's looking like the Rich Club behavior is diminished. So how could we predict that's going to happen from the baseline behavior is the challenge. We took these curves and we took a read off of this point here. This point here is telling us the maximum value that this normalized Rich Club coefficient reaches. And that can be thought of as a measure of the prominence of this club. The higher that point is, the denser the connections between this club. The lower it is, the less dense they are. So we read that off as a measurement of how prominent is your red club from individuals. We then look at the population level, see what we find in each group and across time, and find that the maximum red club coefficient changes over time in TIA patients, but not in controls. So we find that it decreases in the TIA group as time goes on, but again, not in controls. So it doesn't seem to just be healthy aging. It's a small effect, 0.082, but it is an effect that we see that over time the TIA group reduces. Interestingly though, cognition does not differ longitudinally. So these groups, you still can't find any behavioral differences in terms of a various battery of cognitive tests. It's entirely subclinical still, but we do potentially still see this difference in the red club coefficient before this arrives. And we know this is a group where a greater proportion of them are going to go on to develop cognitive decline. So if we remember back to how I mentioned at the start of what we know about healthy brain architecture, we can think about what this means for brain dynamics in terms of if we have a given wiring, a given structure, dynamics then exists on this wiring. This wiring is what facilitates these dynamics, it's what constrains them. And we can imagine it as creating a kind of energy landscape where some brain states are easy to realize, some brain states are more challenging to realize. There may be different paths through this landscape that are easier to take, some that are harder to take. And trying to map out this landscape is part of the aims of network control theory. Given our findings on the red club and disruptions to this architecture, we start wondering about what does this then mean for this energy landscape. So there's a couple metrics we can use to go after this. Average controllability and modal controllability. Average controllability indirectly has a tight relation with these red club nodes. Regions that have a high hubness, a high degree, also tend to have a high average controllability. So what we mean by average controllability are nodes that are capable of driving the brain into states that are quote-unquote easy to reach and commonly realized by the brain. There is an overrepresentation of nodes with high average controllability in the default mode. It also characterizes a lot of the red club nodes. Like I said, there's a tight relation with degree. Conversely, modal controllability captures the ability of a particular brain region to drive the brain into more specialized states that are thought to require more cognitive effort and more energy. And these may be more likely to impact brain function and the type of tests that we would go after, as these are greater cognitive efforts, I guess. So once we measured these metrics, we can look at them in 3 different scales. We can look at them across the entire brain, of their ability to control it and drive the brain into different states, the easy to reach or the hard to reach. We can group by the functional networks, or we can continue to just keep it on an average level, on a nodal level, and then pick out the nodes that are particularly good at driving into the easier or the harder to reach states. Statistics, we used a 2-sided Mann-Whitney U-test, linear mixed effect model with a bump for any correction. Comparing the 2 groups, the control and the TIA group, we found the modal controllability across the whole brain, so averaged across all nodes, was lower in the TIA group. It didn't change with age. We didn't find a factor for this, but we just observed the difference in modal controllability between TIA and controls. This may then have a relation between TIA groups' ability to take on higher, more demanding cognitive tasks later on. We also found that on a subnetwork level, modal controllability decreased specifically in the frontoparietal subnetwork as people age. And this is in both groups, TIA and controls, as teasing apart the effects of healthy aging in TIA is important and challenging, and we find controllability has a component in each. TIA group, they have lower modal controllability generally, and this did continue to persist through aging. And then on the level of nodes, this is a preliminary analysis, but we've been able to retrieve a couple of nodes from the average control side and the modal control side that we call these control hubs that appear to be big drivers of the easy to reach states and the hard to reach states. We were able to pick out a region in the occipital gyrus for an average control that has lower controllability in the TIA group, and an area in the insular gyrus that has reduced modal controllability in the TIA group and as age increases. So overall, we're finding that the rich club prominence is altered in TIA patients and before any sign of cognitive decline, which is exciting to see a network change that we may be able to quantify before anything is clinically measurable. We're also getting some early results that controllability is diminished in a few contexts in TIA patients, on the level of the whole brain, on the level of specific functional networks and the frontal parietal network, and on the level of a couple of specific control hubs. And as we learn more about this may give us some clues into perhaps a more mechanistic relation between the effects of TIA and cognitive abilities. So I want to then bring a bunch of that together for network control, and what can network control do then as a framework to explain some of what we're seeing and to give us hope to find ways to predict interventions. So to do that, just motivate a little with TMS as a specific example. So transcranial magnetic stimulation is approved for treatment of treatment-resistant depression, for example, where we literally apply a magnetic pulse to the skull and hope that this then has a therapeutic effect to depression. My understanding is that the way this is done is literally with placing fingers on the brain and then zapping where fingers are. But we are so different. We are all so individual, and I've been talking to a few of you about grappling with this. We're so individual that from the network point of view, our functional connectivity is as unique as our fingerprints. People can come back to the scanner months or years later and you can identify who it was from the previous time. So why are we using a one-size-fits-all approach for everything when we need something so individualized? And something like network control theory is one inroad to allow us to potentially do this that I find exciting. If we're able to define this energy landscape that I've alluded to for each individual, which is then defined by our individual network, we can find individual ways to then traverse this landscape and think about how to move around it and how to kick people in particular directions if necessary through this landscape. And so in a study that I did with colleagues in Boston a few years ago, we took a sample of subjects from the Human Connectome project, and we created highly individual networks and highly individual atlases from their neuroimaging data. So individual anatomy, individual parcellation even, individual structural connectome, individual graphs. Of course, when you look over everyone, you see things that are very qualitatively similar. Good. We see the same kind of split between hemispheres, less connected cerebellum, similar regions for hub nodes, etc. But when you zoom in and look at these individual connections, you see a lot of differences, and you start to see how these show up in individuals. We then set out to run a number of simulations where we begin with a baseline state, which is something we just extract from data. So we extract this from fMRI time series. And then we try to reach some target state with our simulation. So this target state, it could be anything that we wanted. We set out to use target states as each individual functional network that we've described before, so just depicted with ones and zeros. So with the network being perfectly activated as ones in these regions and 0 in all other regions. So this one network activated, everything else inactivated. So then how do we get from A to B? So we've got the observation, we've got data, we want to drive the brain to some final state. We simulate this and apply network control theory to understand the optimal transition from A to B. And this is a computational model for the simulated TMS pulse. Then what we get out of this is something like the following. So if we've got some initial state and we're trying to get to some target state, you can arrive at this state via a number of different ways. Some of them are smoother than others. Some of them arrive closer to your desired target state than others. But we can move it in this direction. We compared a number of different ways of arriving at this target state. The first one was what we're calling traditional, so from the literature and clinical practice, which regions would you stimulate if you wanted to activate this subnetwork? Then personalized, so this is where we use the network structure, but we keep it simple. So we do a measurement of path length, so just which node is closest to your target state to try and stimulate the entire target state. And then finally, network control theory, so bringing in a more complex methodology of if we're going to take this approach, so modeling it as a dynamical system and just what's the optimal trajectory from A to B, how do I arrive at this, and then compare the three approaches. So the first one is practice, the next two are network level, with one of them being as simple as it could get, what's path length, and then the final one taking the network control theory approach. Okay, and what we found, this is an example of trying to activate the default mode network. The top line here, so again this is the traditional, and then the bottom two are the network. All of them activate a bunch of the target network, but we find that the network control theory approach does so faster, and it seems to do so via a group of nodes in temporal regions, and it more completely manages to activate the network more completely than the other two approaches. A little more on the same thing here. So these ones in orange and red, these are traditional approaches. When it comes to TMS, we can comfortably stimulate two regions at once, and computationally we can do largely anything. So we went with what would be helpful in practice. We can do up to two regions. So this is just one region, second region in prefrontal cortex and precuneus, that are the top two regions, they're both prefrontal cortex, that are the top two regions that are normally stimulated, and then stimulate them together, so we'd expect that to be more effective. On the y-axis is the number of nodes activated within the top nodes of default mode. So you find that more regions are activated with both as expected, but not a ton more. Then move over to the green, the blue and the green. This then is using path length approaches, and this does a little better than clinical practice, but only marginally so. And then the network control theory approach does better again. In grey, these are null models that are just asking what's the effect of topology, what's the effect of space. So we randomize the network while constraining topology in space. And these perform wholly inadequately compared to pretty much everything, so there's more going on than just this. The diagram on the right is telling us the specificity, so it's all well and good saying I've activated default mode. It's not so good if I've just set the brain on fire, if I've just activated everything. So what's the specificity? It's really good. What we're seeing on the right for network control theory, we see from memory looking at this figure, it's about 96% specificity of default mode compared to the rest of the brain. The next component of this is returning to the individual variation comment. We identified regions that would be optimal for each individual to stimulate their default mode network, and then had a look across subjects, what they look like. In each of these figures, you'll see we've got the optimal target site highlighted as the yellow node, and then highlighted in colors are the fibers that are directly connected to it. These are just some examples of across the subjects of what we found to be the optimal stimulation site. And like I said, they're direct connections. They're really different. You're finding a lot of variation here. If you plot everyone together, you see this kind of scary figure saying this is what difference we're finding between individuals. A smaller yellow node means a smaller number of people have this region predicted to be the optimal one to stimulate default mode. Larger means a greater number of people have this predicted to be the optimal stimulation site. So this is what we're finding on our sample, which, like I said, is really contrasting approach of trying to do the same thing for everyone. A quick aside on a potential impact of this kind of treatment. Some of my colleagues in Boston were studying the effects of meditation on schizophrenia, and they taught people to engage in mindfulness meditation such that they could suppress the activity within their default mode network. They were given a neurofeedback situation where they're lying in the fMRI scanner. If they successfully suppress the activity in default mode, then a little white dot moves in one direction, and if they're unsuccessful, moves the other way. So they learn how to suppress activity in the default mode network through mindfulness meditation. Following this, they experienced several weeks of a reduction in auditory and visual hallucinations. That's amazing. Just for something as simple as meditation to affect activity within our large-scale brain networks. Compared to what some people go through for medication for schizophrenia. We then continued to have a look at, just as a kind of principle of what this could then be applied to. We don't have to limit to functional networks. We could do anything we wanted. So we looked to Neuroscience to identify regions that are associated with various mental illnesses and brain disorders, and applied them firstly to depression. So we picked out brain regions that are associated with depression and set out to try our framework on this. And we found the same things that we were finding for functional subnetworks, that a simple network approach, just using path length, appears to outperform clinical practice. These are all simulations, so this is just evidence of outperforming rather than actually showing it does in practice. That remains outstanding. And then the network control theory approach greatly outperforms path length, and the null models perform largely woefully again. So our simulations predict that the network control framework can provide ways to systematically assess the efficacy of stimulating different target sites to activate whole subnetworks. We predict the network control can identify target sites that greatly outperform other methods, but even simple path length approaches often perform better than current clinical standards. The individual network itself is crucial to take into account. We're doing so much on an average perspective and one size fits all, and we see over and over again that that's not good enough. We hypothesize that trajectories induced by network control predictions are more direct and require less control energy. This is an outstanding piece of work, but a hypothesis we want to test. And potential applications of this include brain disorders like depression, schizophrenia, and for improving cognitive function generally. So I'm about out of time, so I just want to give you a quick teaser of a study that we're launching now that I'm really excited about that brings these themes together. We are talking about trying to identify who is vulnerable to specific pathologies, specific mental illnesses, brain disorders, and diseases. One of the largest risk factors for many of these conditions is preterm birth. It exists from birth. People who are born preterm face an increased risk of autism, ADHD, depression, anxiety, and psychosis. And this makes this a good model system for transdiagnostic vulnerability. We know that a lot of network features, including Rich Club that I've mentioned, they form before birth and then mature and develop after birth, but they exist before birth. We know that preterm birth leads to really highly heterogeneous changes in the white matter connectome, but we don't exactly know how, and we don't exactly know how this relates to risk of different conditions. So if we had high precision data, could we predict which individuals are more at risk of different mental illnesses? And this is a framework that I'm hoping that we'll be able to develop with network control theory, that this energy landscape that our brain connectivity describes, of which states are easier to realize, which ones are harder to realize, this is then disrupted in various different conditions. And if we have a look at this energy landscape, maybe we'll be able to explain why some people find themselves stuck in states that are more associated with conditions, say psychosis, say depression. And perhaps this will allow us to cluster individuals at greater risk. So we're just launching this study called BRISK, Brain Imaging Markers of Risk. I'm starting to collect pilot data now for high precision structural and functional neuroimaging and behavioral data for children born preterm and full term. We'll be creating personalized network models and setting out to identify these structures and states associated with various mental illnesses and predict an individual's vulnerability. So I have lots of people that say thank you to you for all of this work. Shining Chen, she's about to graduate from her master's with me. She did all of the work on ovarian hormones and some of the control work in TIA. Joe Adriano, he collected the ovarian hormone data. Phil and George, they're in the TIA study. Phil is leading it, and George is a very talented undergrad who conducted most of the Rich Club analyses. Ariana is the one who's behind the network control theory and TMS approach with Laszlo Barabasi, Emiliano Santinecci, David Momi, and Antonio Valesi also collaborating on that paper. And Signe Bray, she's the co-PI with me on the BRISK study. So great shout out to these folks. Shining made all of her code available on her GitHub. And if any of you were interested in networks approaches, she's put a lot of effort into trying to make this accessible to interdisciplinary audiences, to get people off the ground who want to try some of these approaches and don't necessarily know how to. So she's got some great resources on her GitHub. And then thank you to all of my funding sources and my team's funding sources.
Thank you again.